Meinhardt and Winston presents Modern Biology Guided Reading Audio CD Program.
Please open your book to page 4 to the chapter titled The Science of Life. Follow along in your book as you listen to this chapter. Be sure to look carefully at the figures and illustrations when they are mentioned in the reading.
Chapter 1, The Science of Life. You are about to embark on a study of biology, the science of life. As you work through the chapters of this book, you will gain an idea of the enormous scope of the field of biology. In Chapter 1, you will learn about the unifying themes of biology. You will also learn about the methods scientists use to answer questions, about different types of microscopes, and about scientific measurement.
Please look at page 5 to begin section 1-1, Themes of Biology.
The study of life. Scientific evidence suggests that life has existed on Earth for more than 3.5 billion years. The first living things were one-celled organisms floating in the seas, and it is thought that for millions of years, these were the only life forms on Earth. As time passed, new kinds of organisms appeared. Today, plants, animals, and other organisms are found in every corner of our planet, from the ocean's deepest parts to the tops of mountains. Biology is the study of all living things, from single cells to large-scale interactions of millions of organisms. Biology examines the history of all life on Earth, as well as the life of a single organism, such as the one-celled alga shown in Figure 1-1. How, then, do scientists organize their study of such a large subject? Biology is united by a few major themes. These themes come into play no matter what is being studied, from bacteria to brontosauruses. Turn to the next page. Cell structure and function. The first of the 6 themes of biology is that of cell structure and function. Cells are the fundamental units of life, and all living things are composed of one or more cells. Within each cell lies the machinery necessary to carry out all of the basic processes of life. In unicellular or one-celled organisms, a single cell carries out all of the life processes of the organism. Look at the different types of cells shown in Figure 1-2. In multicellular or many-celled organisms, cells undergo differentiation. That is, they take on different characteristics and become specialized. A specialized cell carries out only one of the many jobs necessary for the life of an organism. All cells have certain things in common. They are bound by a membrane, and they carry a set of genetic information that contains the instructions for every aspect of the organism's structure and function. The second theme is that of stability and homeostasis. Think about your own body. Temperature, water content, and other aspects of your body function at a remarkably stable level. This stability of internal conditions in living things is called homeostasis. Both unicellular organisms and multicellular organisms maintain the delicate balance that is homeostasis. Look at the next page. Notice the body of the seal shown in Figure 1-3. The seal must maintain a stable body temperature even as it moves into and out of freezing water. The third theme is that of reproduction and inheritance. The ability to reproduce is a hallmark of living things. Hereditary information, which is necessary for reproduction, is contained in a very large molecule found in all living things called deoxyribonucleic acid, or more commonly, DNA. Figure 1-4 shows DNA from a unicellular organism. The yellow loop is a continuous strand of DNA which contains several different genes. A gene is a segment of DNA that contains the instructions for a single trait of an organism. The DNA in each cell of an organism is the same as the next cell. In other words, each cell of a multicellular organism contains the entire set of information necessary to construct all parts of the organism. However, cells use only the part of the instructions that they need to do their job, ignoring most of the information in their DNA. Reproduction proceeds differently in different organisms. Some organisms reproduce sexually. Hereditary information from two distinct organisms is combined to create a third organism, the offspring. In most animals, an egg from a female joins a sperm cell from a male to produce the first cell of a new organism, one that combines traits of both parents. However, some organisms reproduce asexually. Bacteria and many other organisms produce offspring by dividing in half, which produces two organisms, each an exact copy of the parent. Turn to the next page. The fourth theme is that of evolution. The enormous variety of organisms today is the result of many millions of years of evolution. The term evolution means change. Organisms change over time as a response to demands that their environments make on them. The most important force that drives evolution is a process called natural selection. The theory of evolution by natural selection is based on the idea that within single types of organisms there are differences among individuals. Those individuals that have traits that help them cope with their environment tend to have a higher survival rate. They leave more offspring than individuals that don't cope as well. As a result, inherited traits that help an organism survive will increase over generations, and traits that do not help an organism survive will decrease over generations. Look at Figure 1-5 to see a helpful trait that has evolved in the Arctic hare. Look at the next page. The fifth theme is that of the interdependence of species. Organisms do not live in isolation. To understand a living thing, we must study its relationship with other living things and with the environment. This branch of biology is called ecology. The study of ecology recognizes that organisms are very dependent on their environments for food, water, and air. In turn, they affect their environment. Tropical rainforests, such as the one pictured in Figure 1-6, contain many different species of organisms. But rainforests are fragile environments on thin soil. In recent years, large areas of tropical rainforest have been cut down or burned by humans. Plant and animal species disappear, and the soil is washed away and becomes hard, which makes the land unable to support further plant growth. Turn to the next page. The sixth theme is that of matter, energy, and organization. Although living things are composed of the same materials as non-living things, living things have highly organized structures that require a constant supply of energy. Our sun provides almost all energy used on Earth. The process of photosynthesis in plants and other organisms converts the sun's energy to chemical energy. Humans and other organisms get this energy from consuming these photosynthetic organisms. Organisms that make food, such as plants, are called autotrophs. Organisms that get energy by taking in food are called heterotrophs. Heterotrophs, such as animals, may consume autotrophs or they may consume other heterotrophs. Figure 1-7 shows salt, which is found in both living and non-living things. In living things, substances such as salt become part of the organism and are used in complex chemical reactions. While non-living things have structural organization, living things are far more complex than non-living things. This complexity and organization are hallmarks of life.
Look at page 11 to begin section 1-2. The world of biology.
Characteristics of life. All living things share certain characteristics. These characteristics include a body made of one or many cells, an organized structure, use of energy, maintenance of homeostasis, growth, and reproduction. Let's consider the first of these characteristics, cells. All living things are composed of one or more cells. In multicellular organisms, cells are specialized for different functions. For example, Figure 1-8 shows the cross-section of a plant stem. You can see different structures, each composed of different kinds of cells. Related to cellular structure is the organization of living things. Looking again at Figure 1-8, you can see that the different structures in the plant stem are arranged in an orderly fashion. Cells and groups of cells are organized by their function in the plant stem, as they are in every multicellular organism. Turn to the next page. Many different chemical processes use energy in an organism. The total of all these processes is referred to as metabolism. Energy is required not only to grow and reproduce, but also to maintain the order and organization of substances that make up the bodies of living things. Some of the energy taken in by an organism is used to maintain homeostasis, or a stable internal environment. Even unicellular organisms strive to achieve homeostasis. In Figure 1-9, you see a bird that has several strategies for maintaining its body temperature at an even level. Some non-living things, such as icicles, grow by accumulating more of what they are composed of. Living things grow too, but they do so primarily by cell division. In cell division, one cell divides to become two cells. Each of these cells divides to produce 4 cells, and so on. Figure 1-10 shows cell division in a unicellular organism. Development of an adult organism results from cell division and a process called differentiation. During differentiation, cells become different from one another so that they can perform different tasks in the body. Look at the next page. All living things reproduce. That is, they produce offspring like themselves. Because individual organisms do not live forever, reproduction ensures that a type of organism will continue on. In many organisms, genetic information from 2 parents is combined in a single offspring, a process called sexual reproduction. Look at Figure 1-11. A small frog has laid many eggs. Each egg has the potential to become a new individual. Though this frog will die, its offspring and their offspring will continue the species. The living world. The world of living things is so enormous and varied that scientists are still exploring it. New species are discovered every day. Many species are too small to be seen with the naked eye. For example, a single gram of fertile soil may contain as many as 2.5 billion organisms. It's easy to see why there is still much to be learned about life.
Please look at page 14 to begin section 1-3, Scientific Methods.
Scientists use certain methods to answer questions. Information is gathered and analyzed in specific steps that are universal in science. These methods of gathering and processing information in logical and organized ways is referred to as scientific methods. Observing. To illustrate the use of scientific methods, we will consider a serious problem that occurred several years ago and the way that scientists worked to solve it. In 1976, a deadly epidemic with an unknown cause swept through several villages and towns in what was then called Zaire in Central Africa. The symptoms of the disease were horrible. An affected person bled internally and from the skin and openings of the body. Death came rapidly from shock, which is the collapse of the cardiovascular system. The disease came to be known as Ebola fever, after the Ebola River, where many of the villages that were stricken are located. Asking a question. All scientific investigations must start with a question. The most important question in the early stages of this epidemic was, what is causing this disease? Look at the next page. Collecting data. The word data refers to information. In a scientific investigation, information about many aspects of the problem is collected. Data collection typically begins with observation. You are familiar with observation. You do it all the time. You are observing when you use one or more of your 5 senses to take in information. Figure 1-12 shows a scientist observing something very small with the aid of a microscope. The second aspect of data collection is measuring. When we measure something, we might determine how large something is, how heavy something is, how many of something there are, and so on. Let's consider measuring as it applies to the Ebola fever scientific investigation. First, scientists counted the number of people with the disease. Then they determined how victims of the disease died and how many days a victim was ill before he or she died. The scientists made other measurements that would help them form a picture of the disease and how it spread. Turn to the next page. The third aspect is called sampling. A sample is a small part that represents a larger whole of something. For example, if you wanted to know the overall opinion in your school about a certain issue, you might ask 20 randomly chosen people what they think about that issue. A sample gives a scientist clues about what is occurring with the whole population. Scientists in Zaire took many blood samples from both sick people and well people. These samples helped the scientists determine what caused the disease. Look at figure 1-13. This is a photograph taken through a microscope of a virus that was found in blood samples from infected people. The fourth aspect of data collection is organizing data. As scientists work to gather information, they also organize it so that it tells them a story. For example, to understand how Ebola fever was spread, scientists determined how sick people and well people had come in contact with one another. The chart shown in figure 1-14 is an example of how scientists organized data to determine how the disease was spread. Look at the next page. Hypothesizing. After scientists have gathered and organized their data, they try to suggest a possible explanation for what they have seen. This explanation, which is called a hypothesis, must explain their observations and must be testable. Let's consider the process of forming a hypothesis. The most important hypothesis in the investigation of Ebola fever concerns the cause of the disease. When scientists found the strange-looking virus in the blood of some people, they hypothesized that the virus caused the terrible disease. This statement explains the cause of the disease and can be tested. If testing a hypothesis shows it to be false, the hypothesis must be discarded. On the other hand, a hypothesis cannot be proven true. A hypothesis that seems true after all evidence is weighed is said to be supported by the evidence. To test a hypothesis, one makes a prediction. A prediction often takes the form of an if-then statement. For example, in the case of Ebola fever, scientists predicted that if the strange virus caused the disease, then putting the virus into a healthy animal would cause the animal to have the disease. Experimenting. Scientists test a hypothesis by performing a controlled experiment. In this type of experiment, a comparison is made between two groups, a control group and an experimental group. These groups are identical except for one factor. Turn to the next page. The differing factor is called the independent variable. During the experiment, a scientist observes and measures another factor called the dependent variable in both the control and experimental groups. The dependent variable is so named because its value is determined by the independent variable. In the investigation of Ebola fever, a controlled experiment was conducted. Test tubes containing healthy cells from monkeys were divided into two groups. In the control group, nothing was done to the test tubes. In the experimental group, blood from infected people was added to the cells in the test tubes. The dependent variable was the health of the cells in both groups of test tubes. Data collected during an experiment must be analyzed. Scientists must determine if the data are reliable and what the data mean. Do they support a hypothesis? If they do not, do they suggest another hypothesis? In the study of the Ebola disease, scientists observed that cells in the test tubes to which the infected blood was added died, while cells in the other test tubes remained healthy. Drawing conclusions. The final step of a scientific investigation is to produce a model. In experimental science, the word model means an explanation of the data. In this investigation, scientists tried to formulate a model of disease transmission. Look at the next page. The scientists' model of Ebola transmission noted that the virus is transmitted by close contact with an infected person. The scientists concluded that the disease spread very quickly under crowded, unsanitary conditions, such as those found in hospitals in poor villages, as Figure 1-15 shows. Parts of the model are incomplete, however. For example, it is not clear how the virus is spread to humans, or if it is carried by animals in the wild. When we draw a conclusion that is not based on direct observations, we are inferring. For example, if you see smoke, you may infer that its source is a fire. Although inferences are not directly testable, they are considered an important part of scientific investigation. In the Ebola fever investigation, scientists inferred that the disease-causing virus is probably carried by animals that occasionally come into contact with humans. The conclusions drawn from scientific research may be used to form a theory. A theory is not the wild guess that many people think it is. Rather, a theory is a broad statement of what is thought to be true, based on extensive experimental evidence and the testing and evaluation of many related hypotheses. Turn to the next page. Implementing scientific methods. Scientific methods can be tailored to fit any type of investigation. Different kinds of scientists conducting different investigations may choose and combine different scientific methods to best fit a particular problem. For example, a field biologist investigating a possible animal source of the Ebola virus might combine observing, sampling, organizing and analyzing data, and hypothesizing. A public health official concerned with the spread of Ebola might combine sampling, organizing and analyzing data, predicting and inferring, and so on. Communication among scientists is another very important part of scientific investigation. Figure 1-16 shows a scientist presenting his research at a scientific meeting. Scientists build on the work of other scientists, and all scientific work is open for criticism or verification by other scientists. Communication is particularly essential in the case of a serious disease such as Ebola. Through cooperation and communication, many lives that would have been lost were spared from this disease.
Please look at page 21 to begin section 1-4, Microscopy and Measurement.
Much of biological research involves observing things that are very small. Biologists use special tools called microscopes for this task. Microscopes. A microscope is an instrument that allows us to see things that are very small by producing an enlarged image of an object. The increase of an image's apparent size is called magnification. The power to show detail refers to a microscope's resolution. One type of microscope is the light microscope. Figure 1-17 shows a drawing of a compound light microscope, the most common kind of microscope used in biology. Look for a moment at the different parts of the light microscope shown in the figure. A thin glass slide holds the specimen to be viewed. The specimen must be very small, such as a bacterial cell, or it must be sliced very thin to allow light to pass through it. The slide rests on the stage of the microscope. Directly below the stage is a light source, either a mirror or, as in this case, an electric light bulb. Light passes up through the specimen and into the lenses of the microscope. The first lens the light encounters is one of the objective lenses located on a revolving dial called the nosepiece. The image of the specimen is projected up through the body tube of the microscope to the ocular lens. An observer looks down into the eyepiece to see a magnified view of the specimen. Turn to the next page. On most light microscopes, there is a set of objective lenses with differing powers of magnification. The factor of enlargement of an image is called the microscope's power of magnification. You can compute the power of magnification of a microscope by multiplying two values. The power of magnification of the ocular lens, which often is 10 power, is multiplied by the power of magnification of the strongest objective lens. In many school laboratory microscopes, the strongest objective lens is 40 power. So, the power of magnification is 10 times 40, or 400 power, which is written 400 X. Let's consider a different and more powerful microscope, the electron microscope. At magnifications greater than 2,000 power, images become blurry with the light microscope. Scientists who wish to examine very small specimens, such as viruses or parts of cells, might use an electron microscope instead. In an electron microscope, a beam of electrons, rather than a beam of light, produces the image of a specimen. Look at the two specimens shown in Figure 1-18. They are the unicellular organism called Giardia. The upper photograph was taken using a transmission electron microscope, which is abbreviated as TEM. In a transmission electron microscope, a beam of electrons passes through a thinly sliced specimen. Magnetic lenses focus the beam on a photographic plate. Transmission electron microscopes can magnify images up to 200,000 times. The lower photograph was taken using a scanning electron microscope, abbreviated SEM. This kind of microscope shows surface details of a specimen. The specimen must be prepared by spraying it with a thin metal coating. An electron beam passed over the specimen results in a shower of electrons which are projected onto a photographic plate. A scanning electron microscope can magnify images up to 100,000 times. Look at the next page. Measurement. All scientists use a single standard measurement system called the International System of Measurements, or SI, which is based on a fundamental set of measurements called base units. Table 1-1 shows the 7 base units of SI. The base units describe length, mass, time, and so on. Base units can be multiplied by powers of 10. The prefixes that are attached to the base units are shown in Table 1-2. Turn to the next page. There are also derived units. Derived units are produced by manipulating 2 base units or a base unit and another derived unit. Derived units that are often used in the study of biology are shown in Table 1-3. Still other units, though not part of the SI system, are acceptable for use with SI units. These other units appear in Table 1-4.